Hello to all my dear students. I am Deepen Singh. Welcome to all of you in our school SJEC e-learning YouTube channel. So student, this is your biology class and we are discussing second unit of our syllabus, genetic and evolution. And in this unit we are discussing fifth chapter, principles of inheritance and variation. And in this chapter we already discussed various important processes and concepts, topics related to genetics, such as inheritance and variation, reasons behind the success of Mendel, the laws suggested by Gregor John Mendel, sex determination, and in today's class we are going to discuss about sex linkage or sex linked inheritance. Means the sex chromosome or X chromosome carries genes for sex determination and also some genes for some somatic characters. The genes for somatic characters present on sex chromosomes are called sex linked genes. Means the character which are controlled by the sex linked genes are called sex linked characters, and their inheritance is sex linked inheritance or sex linkage. The inheritance of sex linked character is linked to the sex. Let's see the sex linked inheritance found in Drosophila. First of all, the sex linked inheritance was discovered by Morgan in Drosophila melanogaster when he found sudden appearance of a white-eyed Drosophila and followed its inheritance, and also suggest three crosses. These are the first cross between red-eyed female and white-eyed male. Morgan in 1910 noted a white-eyed male in a culture of red-eyed Drosophila. Means when all the Drosophila have red eyes in color, then suddenly he found that there are some Drosophila also present with white-eyed. Then this white-eyed male was mated with a homozygous red-eyed female Drosophila. Then all males and females of F1 progeny were red-eyed. Let's see the cross. In this cross, the red-eyed Drosophila cross with white-eyed Drosophila, in which red eye represented by female and white eye represented by male character. When these F1 males and females were allowed to interbreed, the F2 generation or progeny consisted following females and male with following character, such as in female all the individuals are red in color, such as homozygous female 25% and heterozygous female also 25%, while in male there are some male, about 25% are red-eyed in color, while remaining 25% males are present with white-eyed color. Thus, this cross represent the homozygous and recessive character. Red eye, which is homozygous character, is dominant over recessive character. In cases such as in the case of male and female. Then Morgan made second cross between F1 red-eyed female and a white-eyed male Drosophila. Morgan made a cross between F1 female, which are heterozygous, and white-eyed male. Then the F2 results or F2 generation were white and red-eyed females and red and white-eyed males in equal proportion. Means there are female present with white-eyed colors are 25% and 25% females are with red-eyed. Then the male are also 25% are red-eyed and remaining 25% are with white-eyed color. Clear? It means in this case both character, red-eyed and white-eyed character, are common and equal in both gametes, male and female gametes. Next cross Morgan made between F2 white-eyed female, which is represented by XW and a red-eyed male represented by X and Y. In F2 generation, all females were red-eyed represented by XW and X, and all males were white-eyed represented by XW and Y. As we can see in the cross, there are the cross made between F2 progeny. White-eyed character represent female gamete and red-eyed recessive character was represented by male character. Then the results are found, means 25% are red-eyed and remaining 25% are white-eyed in both the cases, male and female. And with the help of these crosses, Mendel proposed their appearance or their suggestions about the sex-linked inheritance found in Drosophila melanogaster. The next topic, which is criss-cross inheritance, also suggested by Morgan on the basis of above cross, means the cross first and cross third are reciprocal, and according to Mendel's law, the results of reciprocal crosses are expected to be identical, but it is not so in these cases. Then Morgan observed that in first cross only red eyes appeared in both female and male Drosophila, but in cross third all the females and males were white-eyed. Means from all the three crosses it is proved that the gene for the eye color is located on the X chromosome, while the male transmits its X chromosome to his daughters and females transmit its X chromosome to both sons and daughters. This type of inheritance is known as criss-cross inheritance. Clear? Now, what is the significance of criss-cross inheritance in the Drosophila or in the genetics? First of all, criss-cross inheritance help in establishing relationship between genes and sex chromosomes. Means it provide evidence that sex linked genes are located on X chromosome, and it helps in understanding mechanism of inheritance of sex chromosome and related sex linked disorders. Now, what are the characteristics of sex linked inheritance? First of all, the sex linked inheritance for genes are located on X chromosome, and they are also called sex linked genes or X linked or Y linked genes. And in this way, the Y chromosome is without gene for eye color. The next character is female has XX chromosome, means the XX proportion XX chromosome, and therefore has two genes for each sex linked characters such as eye color. Next, the male has XY sex chromosome, and in this X chromosome is without corresponding allele of eye color gene. Therefore, male possesses only one gene for eye color. Next point is the male produces two types of male gametes. Half of them have X chromosome, means with sex linked genes, and other half Y chromosomes without sex linked gene, means the female produces only one type of eggs, all with one X chromosome and with X linked gene. Next and last point is the red-eyed male offspring of F1 generation transmit X chromosome to F2 females and Y chromosome to F2 male. Therefore, all F2 female receive W gene from male and are all red-eyed. Let's see the difference between autosomal and sex linked genes. As we know, in human being there are 23 pairs of chromosomes are found, in which starting from 1 to 22 pairs chromosomes are known as autosomal chromosomes, while the remaining 23rd pair of chromosome is known as sex chromosome. And on the basis of these, the genes are also divided into two types: autosomal genes and sex linked genes. First of all, autosomal genes are present only on autosomes, means autosomal chromosomes, and the sex linked genes are present only on the sex chromosomes. The next difference is, in autosomal gene, each individual, male or female, has one pair of alleles of each autosomal gene, while in the case of sex linked genes, the male possesses only one allele of sex linked gene, while female has one pair. And Autosomal gene in hemizygous condition is not found in the case of autosomal genes, while in the case of sex-linked genes, males are hemizygous. There are reciprocal crosses produce the same results in autosomal genes, while the reciprocal crosses produce different results in sex-linked genes. Now the topic is variation, means there are also some types of differences present in our body which are also transmit during the cell division. First of all, what is variation? Variation are morphological, physiological, cytological and behavioral differences among individuals of a species and also the offspring of the same parents. Means variation are the rule of nature. No two individuals are identical. Even identical twins when placed in different environment shows differences. Now the next and important topic is types of variation. Means variations are variously classified on the basis of nature of the character influenced, such as morphological, physiological or behavioral, on the hereditary nature of the character as non-heritable or heritable, or the type of cells or tissues influenced as germinal or blastogenic variations. The next topic is there are two types of variation: non-heritable or somatic variation, heritable or genetic variation. Let's see first heritable variation, means genetic variation, means germinal variation, those variation that are passed on to the offspring, and these arises due to the changes in the genetic material, genes of DNA of germplasm or commonly known as gametes. So student, therefore these variations are called genetic variation, and only these variation have evolutionary significance, such as hemophilia, color blindness, blood groups, baldness and intelligence are the example of heritable variations. Then we can say that the heritable variation are of following two types: continuous variation and discontinuous variation. Let's see what are the continuous variation. First of all, continuous variations arise in sexually reproducing organism at the time of gamete formation due to the reshuffling, remixing of the parental genes, producing new combination or recombination of character in the offspring. And the reshuffling of genes may be the following types. First of all, independent assortment, means the random separation of paternal and maternal chromosome occur during the meiosis. For example, random combination of maternal and paternal chromosomes in fruit fly with 4 pairs of chromosomes can produce 16 possible combination. Out of these, 7 pair of chromosomes of garden pea can assort in 128 ways and 23 pair of chromosomes in human being. Clear? The next type is reciprocal recombination. In this, the linked genes occurred by crossing over or mutual exchange of corresponding segments of non-sister chromatid of homologous chromosome during the prophase first or meiosis. This changes number of dominant and recessive genes in the offspring. Next and last type of continuous variation is random fusion. Means the random fusion of gametes at the time of fertilization. Such as there are 4 types of gametes formed from one parent, means having 2 pair of chromosomes. Combining these characters, these gametes with 4 types of gametes from other parents produce 16 types of offspring. It means union of sperm and ova can produce possible combination, and these recombination far exceed human population. For this reason, no two human beings are exactly alike except identical twins. Now the next type of variation is discontinuous variation. Means the variation, the differences in a characteristic that can be observed in a population, characteristics that are determined by the different alleles at a single locus show discontinuous variation, such as garden peas are either wrinkled or smooth. These types of variation are caused due to following 3 types. Means the discontinuous variation are divided into following 3 types of variations or mutations. Mutations means sudden changement in the DNA or genes. These are gene mutation, means sudden changes in the structure of genes. Chromosomal aberrations, sudden changes in the structure of chromosome. And the genomic mutation means sudden changes in the number of chromosomes, known as genomic mutation. There are also some type of non-heritable or somatic variation in which the variation occur in the somatic cells of individual due to the influence of environment, and these variations are acquired by the individual cell during its lifetime and are lost with its death. Therefore, somatic variations are called acquired variations, means they are not transmitted to the offspring, hence are also called non-heritable variations. Somatic variation arise due to environmental changes, use or disuse of an organ or a structure. Clear? So we start the all 3 types of mutation, such as gene mutation, chromosomal aberration and genomic mutation, in our next video. So student, if you like our way of teaching, hard work, so please subscribe us to learn more and be updated about your syllabus. Thank you for watching the video.